The old saying goes, you can't beat Mother Nature.
You know, every time we think that we can beat Mother Nature, we are humbled over and over again.
In cell biology, DNA is usually the star of the show. But DNA is just a blueprint, a set of instructions for building proteins. Now, I know when you hear the word protein, you're thinking, should I have the chicken or the fish? But biochemically speaking, proteins are a lot more than what's for dinner. Enzymes, antibodies, even our muscles are all protein-based.
The research that I do is in biomimetic chemistry. We can either engineer natural proteins or we can engineer synthetic molecules to mimic the shapes, structures, and functions of natural proteins.
With support from the National Science Foundation, chemist Kent Kirschenbaum and a team at New York University are developing new methods for making synthetic proteins in the lab. They see a future, not far off, when they will be able to handcraft proteins that have never existed before in nature and put them to work at the industrial scale.
We've really developed the building block approach to crafting these molecules in an extremely reliable way. We're confident that if we can design a molecule, we're going to be able to build it.
Imagine synthetic antibiotics that could fight infections like MRSA, custom pharmaceuticals to treat advanced prostate cancer, and new enzymes that will turn cellulose into fuel.
One way to think about a protein chain is a bunch of beads on a floppy string.
Inside our cells, biochemical factories build proteins brick by brick, so to speak, the bricks being amino acids. In Kirschenbaum's lab, they've developed methods to do much the same thing on these tiny beads that they load into a syringe.
The beads provide a very nice surface area, and they're also porous so that the chemical reactions can take place on their surface and in the interior of these beads.
The big challenge is working out what's called the protein's folding code, engineering it to naturally fold itself into a precise structure. Shape is everything for proteins. To be effective, they have to fit together with other chemicals like a key going into a lock.
Our job is to figure out what that code is that relates the sequence of those amino acids to its folding property. We think we can crack that code if we go into the lab and synthesize chain molecules with specific sequences of unique, new building blocks.
They check their work using a technique called X-ray crystallography. If the shape of the molecule is even a little off, it won't work as designed. So far, they've had good success synthesizing relatively small ones.
This is a huge, huge deal. We are really at this threshold. What we need to do is take this to the next step so that we're not just mimicking short individual units, but really mimicking the complex three-dimensional arrangements of some of these massive protein structures. Then a whole world of applications will open up for us.
Designing synthetic proteins to create advanced materials, cleaner and more efficient fuels, better drugs, and more effective healthcare treatments. For Science Nation, I'm Miles O'Brien.